There I was, anxiously waiting for my final exam results in Jagdalpur, India. My first grader teacher, Mrs. Panigrai Madam, announced the results one by one. Neha, 95. Mukesh, 70. Rizwan, 63. Wait, where am I? Did I pass? But when she got to me at the end, she said, Piyush, Papa ko bulana, in Hindi, which means call your dad. As a 9-year-old factory worker in India, the idea of finishing the primary school education was just a dream. Every year, my teachers would go down the line of students announcing their results, but they always stopped at me. Why? Because my parents hadn't paid the tuition. You see, money dictated most of my life and my education. I spent hours working as a child laborer after school, and I did the same when my mother started her own papad and chips business or when I started tutoring. Wake up, go to school, work, sleep. That was my day for the rest of my life. And even then, I didn't have enough money to eat or support my family of 4 or for my education, which made me eternally grateful to all my relatives, particularly my uncle, Mr. Jain, who would often pitch in money for our family expenses. As a low-income student in a developing nation, even the healthcare was inaccessible to an unbelievable degree. I was forced to get hospitalized once, all to avoid having to pay for a doctor in the first place. There just wasn't affordable alternatives that could help me or the millions of others living in poverty-riddled conditions. Even when I was young, I knew how important education was. Both my parents weren't educated, and therefore, the education became the center of my life. Well, I thought getting an education could bring me money, just enough to eat and fulfill my lifelong dream of owning a couch, which I thankfully have one now. I wanted to fix my family's financial problems so that my kids wouldn't face the same. So why am I telling you all this? Unlike other scientists, my motivation and path to science was quite unusual. I was fortunate enough that my quest for financial stability through education introduced me to this magnificent field of gene editing called CRISPR that I could use to help the world in the middle of the pandemic. With the support of my family, my relatives, my community, and Times of India, a leading newspaper that gave me scholarship, I somehow managed to come to the US to pursue my PhD right after my undergraduate degree in pharmacy. Under my advisor, Professor Simon Friedman, I explored the world of light-controlled biological processes, literally using light to deliver drugs that could save lives. Using photochemistry, I created a light-activatable insulin depot that you can inject under the skin and can release tiny doses of insulin in response to light, far different than manual methods used by diabetics today. However, my current scientific calling came a few years later at MIT, where I was introduced to CRISPR. So what's all the buzz about CRISPR? I promise you it's more than just a buzzword. With CRISPR, we can literally reprogram any DNA sequence and treat genetic diseases, which was never possible before. With CRISPR, I very quickly realized that I was in this age of revolutionary biological technology that could save lives. So what exactly is CRISPR-Cas? It's like a little surgeon holding a scalpel that can make precise cuts in the DNA. So where does it come from? Just like we get flu from the viruses, bacteria can also get infected with the viruses. We have a complex immune system with many cells to fight against the viruses, whereas bacteria is a unicellular microorganism. Interestingly, bacteria has evolved a unique mechanism called CRISPR-Cas to recognize these foreign viral invaders and cut their DNA into pieces. Scientists realized that this CRISPR-Cas system can be brought and easily be reprogrammed to cut and recognize any DNA sequence. Using CRISPR-Cas systems, scientists and doctors can now repair and replace genes with precision that was never possible before. During my postdoc position at MIT in Professor Sangeeta Bhatia's lab, I had this unique privilege of combining my previous work in photochemistry with my newfound passion for CRISPR. I created a light activatable CRISPR-Cas system, one that allows scientists to pinpoint in space and time which cells we want to gene edit and when. Using such light-based tools, scientists can now turn on CRISPR-Cas system in specific tissues and cell types, such as your skin, eyes, tumor. Now, what I realized is that these CRISPR-Cas systems can not only allow you to target specific tissue types, but also make the gene editing technology safer to use. Where there are many types of CRISPR-Cas systems that exist in nature, and my lab focuses on a special type of CRISPR-Cas known as CRISPR-Cas12 systems. These systems initially work like a scalpel in surgeon's hand, but then the scalpel quickly transforms itself into an automated chainsaw, shredding every piece of DNA that comes along its way. While these systems may not be suitable for gene editing applications, when you need precise cut in the DNA inside the cell, scientists have realized a special use for them in diagnostics by harnessing this unusual power of shedding DNA into amplification of signal. Most common drugs work by binding to the molecules of the cells. CRISPR, on the other hand, not only need to reach to the cell, but also inside the nucleus where our DNA is stored. What further complicates things is that CRISPR-Cas molecules are large in size, and our cells don't give entry access permit to these large freights. So what I started at MIT and continued here in my new lab was to help create a delivery technology by packaging CRISPR-Cas components into nanoparticles such that they can target desirable cell types. It's like FedEx for cells, but with a special entry access permit. Now, I told you about the CRISPR-Cas12 systems, which can be harnessed for diagnostics. And my lab is using these CRISPR-Cas systems for COVID-19 diagnostics. And if you haven't heard about COVID-19, it's a disease caused by SARS-CoV-2, which is a coronavirus. The CRISPR-Cas systems can be easily reprogrammed to find the genetic material of a coronavirus and cut that coronavirus into pieces. Now, when the coronavirus genetic material is recognized by CRISPR, it turns itself into an unstoppable chainsaw, which shreds every piece of DNA that's present in the sample. Now, to visualize these signals, we add millions of DNA reporter molecules that get also chopped up by these CRISPR-Cas systems. When these reporters get chopped up, they light up the sample. And this tells us that the coronavirus is present in our sample. The primary function of the CRISPR-Cas is definitely to detect the coronavirus, but the natural occurring CRISPR-Cas systems are not sensitive enough on their own and often require the use of other amplification technologies. This increases complexity, rendering them handicapped for use outside the lab. So my lab here at UF is engineering the CRISPR-Cas system itself such that it increases its speed and improves the sensitivity of the test. Using engineered tests, we have already developed tests for HIV, HCV, malaria, and even prostate cancer. Now, to curb any pandemic in which the naturally occurring variants are expected, as we are seeing today, we must develop a rapid at-home test kit that can not only detect the presence of coronavirus, but also tell you the variant at the convenience of your home without needing to go to a hospital. And my lab has found such solution. The Food and Drug Administration has already given emergency use authorization to several at-home sample collection kits, antigen detection kits, and even genetic testing kits. While the FDA continues to caution the reliability of these kits, even though we keep getting them in our mails, I believe that the CRISPR-Cas-based systems can be much more rapid and accurate. In fact, the FDA has already given emergency use authorization to 2 CRISPR-based tests. And these tests can be conducted in a high complexity lab environment and has already reached millions. However, what I am interested in is how we can engineer these tests so that they can be conducted at home and can reach billions. The engineered CRISPR-Cas systems can be very accurate and can give an answer within 10 to 15 minutes. They work like a pregnancy test and can show 2 purple colored lines if a test is positive. Using engineered tests, I envision developing a test that would allow detection of any pathogen within minutes without needing an equipment in your saliva sample, urine sample, blood sample. The engineered CRISPR-Cas test can be monitored by your cell phone and can even be tracked by smart wearable devices. The potential for such tests is just the tip of the iceberg, because you can not only apply these tests for detecting any pathogen, but also for genetic diseases, for cancer. Well, the engineered tests can also not only reduce the risk of disease incidence and transmission, but also decrease disease severity by allowing quicker interventions, reducing the chances of next pandemic. And my wish is that these tests will be soon accessible throughout the world. In fact, with the support from funding agencies such as the CDC, we are already deploying these tests to countries such as India, Nigeria, Democratic Republic of Congo. These include places such as where I grew up. If the coronavirus or COVID-19 pandemic would have hit when I was a kid, there's no way I would have been able to afford the specified PCR-based test. It's evident by how hard COVID hit India's most vulnerable populations last year. I'm truly grateful to all those who have helped this research, bringing this research to the entire world, including all my students, my mentors, my teachers, my collaborators, including my first grader teacher, Mrs. Panigrahi Madam, who extorted my exam results. By the way, you're all dying to know, I'm sure, I did pass my first grade with flying colors. Thank you.